AS Level Biology 9700. Paper 2, Variant 1, October November 2024, question number 1 and 2. And question number 1 is animal cell, plant cells, and prokaryotic cells have similarities and differences in their structures. Table 1.1 lists 5 organelles found in cells. And the first row has been completed for this, and we have to take the organelle which is present in these cells. The first row has been completed for you. You can see the nucleus is present in animal cells, in plant cells, but it is not present in prokaryotic cells. So prokaryotic cells don't have the membrane bound organelles and a proper nucleus inside. So the second organelle is the large permanent vacuole. It is present uh, in plant cells, not in animal cells, because in animal cells, the small vacuoles are present. Many small vacuoles are present, which are not permanent, but in plant cells, a large permanent vacuole is present. Again, vacuole is absent in prokaryotic cells, endoplasmic reticulums, which act as transporting systems inside the cell and site for ribosomes as well. So they are present both in animal cells and as well as in plant cells, but they are not present in prokaryotic cells. As I have told you, prokaryotic cells don't have the. So Golgi bodies are also present in both types of cells. Again, they are absent in prokaryotic cells, and centrioles are only present. Centriole, you know, helps in cell division. So for in the formation of spindle fibers as well. So they are not present in plant cells and prokaryotic cells again. So it was all about the question. And next question is part B, in which they have shown us our section through a part of an epithelial cell found in the digestive system of an animal. The cell is specialized for absorption of digested food. So here you can see the P and Q has been labeled for us. So we have to write the answer for these labelings, and P are the villi, which are the, you can say, finger-like projections in the small intestine for absorption. So villi or microvilli, we will write over here. So these are for the absorption of digested food. And the shape increases to surface area for more absorption. So the rate of absorption is increased in this way. And second is Q. Q is the mitochondria, which where the aerobic respiration takes place and energy is produced and stored in the mitochondria. So this energy is used for the absorption of digested food, because digested food is absorbed against the concentration by the process of active transport. So energy is needed for that process. So we can say that mitochondria provides energy energy for absorption of digested food, which is absorbed by active transport, that is against the concentration gradient, and energy is needed for it. I always tried my best to write properly because I write on screen. I'll always tell you that I write on screen, so it is little bit difficult. So listen to me, and then you can match my, after listening my writing, you can match both of them. In this way, it will be understanding for you. Now the next question is question number 2. And question number 2 is, in the mammalian circulatory system, red blood cells travel through different types of blood vessels as they pass from the heart to respiring tissues and back to the heart. Shows the type of blood vessels through which red blood cells travel in the circulatory system. And the heart arteries takes away the oxygenated blood from the heart. So to provide this oxygenated blood to different parts of the body, especially to different tissues, and even up to cellular level, the blood is provided. So this distribution of blood, for this distribution of blood, arteries are further divided in arterioles, and then arterioles are further divided into capillaries. And then capillaries are further, you can say, capillaries branch together. So the capillaries join together to, because capillaries are only one cell thick. So exchange of gases takes place over here. Because oxygen has been provided to the body and carbon dioxide enters into the capillaries. So now this deoxygenated blood has to uh, collect from different parts of the body, and then it has to be towards the heart. So capillaries join together to make venules, and venules join together to make veins. So uh, first of all, the arteries divide and then fill capillaries, which are only 1 cm thick, and from capillaries, the blood vessels will join together to make venules, and venules, they will join together to make veins. The part is, water is the main component of blood. It has an important role in transport of substance around the body. Figure 2.2 shows the ionic compound sodium chloride dissolving in water. Figure 2.2 shows the ionic compound sodium chloride dissolving in water. And here you can see a diagram uh, in which they have shown us the water molecules, and they have shown us the chloride ion and sodium ions, the sodium ions and chloride ions. And uh, the water molecules, they have shown us the water molecules. Oxygen has partially negative charge, whereas the hydrogen has the partially positive charge. Partially, you can say the slightly positive They are because they are the they show the polarity. So we can say that water is a polar molecule because it shows the difference in electronegativity between oxygen and hydrogen. So this means it has a partially negative and partially positive charge. So when sodium chloride is added or placed in water, the delta positive end of the water molecule are attracted to the negatively charged chloride ions on the crystal surface. Simultaneously, the delta negative ends of the water molecules are attracted to the positive charged sodium ions. So the water molecules surround the individual ions, forming a shell of water around each ion. So this process is called solvation or hydration, you can say. So the strong attraction between the water molecules and the ions overcomes the electrostatic forces holding the sodium and chlorine ions together in the crystal lattice. So breakdown of the crystal lattice takes place, and we can say that the more water molecules surround the ion and pull on them, the crystal lattice of sodium chloride begins to break down. And then you can say in this way the hydrated ions then move away from the crystal and become dispersed throughout the water. So after this explanation, I'm going to write all these points which I have you can say discussed with you. So first of all, I would like to show you that the water is a polar molecule as it has slightly positive and negative charge. Slightly positive charge on hydrogen ions, on hydrogen, and slightly negative charge on oxygen. So we can write it in this way that water is a polar molecule due to the difference difference between oxygen and nitrogen. As positive and oxygen as partially negative. When sodium chloride NaCl is placed in water, the delta negative of or delta positive of water molecules are attracted towards the chlorine ions and next are attracted towards the sodium ion. The water molecules mm, space is less as I'm writing in bold uh, font, so I'm writing above, but you have to write within this given space. But little due to my handwriting, it is little bit difficult for me as I am writing on the screen. The water molecules surrounds from the individual lines forming a shallow water around them. The strong attraction between water molecules will help to dissolve the NaCl in water. Because the strong attraction between the water molecules and the ions overcomes the electrostatic, you can say the forces which are holding the sodium chloride together in the crystal lattice. So this breakdown of crystal lattice will take place, and as a result of this breakdown, so the sodium chloride begins to break down and it will be completely dissolved into the water. So in this way, sodium chloride is dissolved into the water. I have taken more space again and again. I am telling you, but you have to write within this given space in short font. So the next question is figure 2.3 shows a Galapagos penguin, Stephensis mandiculus, swimming in the water. Penguins are birds that live on land but spend a lot of time swimming underwater hunting for food. Penguins can remain underwater for up to 20 minutes. So they can live for up to 20 minutes. During this time, they do not breathe, but their tissues continue to respire. Hemoglobin in the red blood cells of penguins has a higher affinity for oxygen than hemoglobin in other birds that do not swim underwater. So figure 2.4 shows the oxygen dissociation curve for a bird that does not swim underwater. So draw a line, figure 2.4, to suggest the position of the oxygen dissociation curve for penguin hemoglobin. So here is associate, you can say, a curve, oxygen dissociation curve. So which you can see, and it shows the affinity of the oxygen. So first of all, I will, you can say, explain this. What is this uh, oxygen dissociation curve? So the oxygen dissociation curve, it is, we can say that it is also known as the oxyhemoglobin dissociation curve, which is a graph which shows the relationship between the partial pressure of oxygen in the blood and the percentage of hemoglobin that is saturated with oxygen. In simple terms, it illustrates how readily hemoglobin in red blood cells binds to oxygen at different oxygen concentrations. And you can see its shape. Its shape is a sigmoid shape or S shape. You can see this S shape. This shape reflects the cooperative binding of oxygen to hemoglobin. So, question, I have moved to the question again that we have to draw one. This is the curve for a bird that does not swim underwater, but we have to show the position of the oxygen dissociation for penguin hemoglobin, which can swim underwater for about 20 minutes. So it means it has more affinity for oxygen. So this curve shows the affinity for oxygen. So in this way, we can see that the percentage saturation of hemoglobin with oxygen is more in penguin as comparison to that bird, because the penguin can survive in water for more time, for about 20 minutes. It means it has more affinity, or its hemoglobin has more affinity for oxygen. It means its body has more oxygen as comparison to that bird. So in this case, when the oxygen saturation is high, at that time, this curve is shifted to the left. So we will draw this curve in this way. So uh, let me draw it again, and let me draw it again, as it is little bit, it will move in this way. I'm trying to draw it with smart pen, which is not a good idea. So I'm drawing it with simple. So in this way, it will be shift towards the left, because it has more affinity with oxygen as comparison to that part. And the next question is, penguin hemoglobin is very sensitive to decrease in pH caused by an increase in the carbon dioxide concentration in the blood. Explain how a decrease in pH affects penguin hemoglobin and suggest how this helps in penguin to swim underwater for a long time. So I would like to explain it in this way. That is the carbon dioxide and pH. When carbon dioxide dissolves in blood, it forms a carbonic acid. which then dissociates into hydrogen ions and bicarbonate ions. So an increase in carbon dioxide concentration leads to an increase in hydrogen concentration, thus lowering the pH, making the blood more acidic. So hemoglobin is the protein in red blood cells, which is responsible for carrying oxygen from the lungs to the body's tissues. So when we can say that penguin hemoglobin is very sensitive to a decrease in pH. So when it happens, that is, a decrease in pH means the hydrogen ions concentration is increased. So causes a significant change in hemoglobin's properties. So a decrease in pH generally weakens the bond between hemoglobin and oxygen. This is also known as the Poffer effect. The presence of hydrogen ions competes with oxygen for binding sites on hemoglobin, effectively reducing hemoglobin affinity for oxygen. So in the tissues, this reduces affinity, uh, is beneficial. It facilitates the unloading of oxygen from hemoglobin and delivers it to the tissues that need it most. So in this way, it happens, that is, decrease in pH affects penguin hemoglobin and it helps the penguins to swim underwater for a long time. I'm going to write this explanation right now. So again, I will use the more space to write. So first of all, I will start the presence of, uh, this is a decrease in pH then weakens the bond between oxygen and hemoglobin. Presence of hydrogen ions removal effect. The presence of hydrogen ions competes competes with oxygen for binding sites. On hemoglobin, effectively reducing affinity for oxygen. This reduced affinity is beneficial It it facilitates the unloading of oxygen from from hemoglobin to the respiring cells. That need it most. It helps the penguin to swim underwater. So in this way, it is beneficial for them. You can listen if it is not, and this writing is not understanding for you, then listen to me, my explanation. It will be better helpful for you. Now the next question is, the heart rate the heart rate of a penguin decreases while it is swimming underwater. Heart rate is regulated by a group of specialized cells in the wall of the right atrium. The activity of these cells is modified by nerve impulses. So name the group of specialized cells in the wall of the right atrium that regulates high heart rate. So you must know that that is SAN. So it stands for sinoatrial node. Sinoatrial node. So it is a very important part of our heart because the heart's natural pacemaker. So pacemaker, natural pacemaker. So this is the natural pacemaker. So the sinoatrial node is a tiny cluster of specialized cells located in the upper part of the right atrium, one of the heart's four chambers. So it generates electrical impulses. It generates electrical impulses. So this is all about the question number 1 and 2. Please like, share, and subscribe my channel. Thanks for watching.